You guys are all set to go, so.
Hi, David. I'm gonna have you unmute yourself. All right, perfect. And I'm perfect. I see your screen. David, you are still, there you go. You're not muted.
Okay. Okay, so,
David, I'm gonna make you a co-host, okay?
okay. Yeah.
Yeah, I'm gonna stay with you, so I'm gonna move you on over. I'm gonna add you to the preview right now. Christy has already posted.
Hi, David. It's Richie. Good to see you.
Hi, Richie. Hey, I'm really sorry. I enjoyed listening to the first two talks, but for some reason I can't connect as a presenter.
Yeah, I
Wow.
heard. It's been a little funky, to be honest. So it's the first time we're using this platform.
Yeah, just let people, yeah.
So I'm glad we were able to figure out an
Yeah.
alternative.
Hi, David.
Yeah. Hey, Ned. Yeah, it's been a great session. Great two presentations. I've been I had no problems listening and watching them, but I can't connect to
Well,
present mine.
That's okay. Well, this will work. So
Yeah, this will be fine.
All right.
thanks. Yeah, thanks for doing it, David.
Oh, sure. No, it's been a great morning and looking forward to the rest of the day.
Yeah, it's
We
great to
really
have you.
appreciate it.
So are we not, Rich, Hannah, are you there? Are we, how is this working with all the attendees?
I
Richie?
imagine they're,
Oh.
well, we can ask her.
Yeah, I know she's not responding. I don't know why, but I assume everything's switched over. Hannah or Brooke? Hey, Richie, are you here like a little, oh, there you go.
Yeah,
Hi.
Brooke's here.
Hi. Yeah, I think what they were saying before I moved over to Zoom is just that, yeah, all the attendees are supposed to join. They're going to send a message on the other platform with the Zoom link, and then everyone can just join if they're still on that
I think
other
they're
platform.
joining in now. I think we can sort of see maybe.
Yeah, so I think we should wait another minute. or
Yeah.
two.
You know, we have, we can run over too, so let's not get too, you know.
Yeah, they're all coming in now.
Richie, did you have any sense of what went down with the other platform? Did you hear it?
No. No, it was working fine at that point for me.
It's good to have a backup.
Yep.
We have, I don't know how, over 400 participants. So this could,
They're slowly rolling in.
yeah.
Well, let me just make sure I can get my screen shared appropriately while we're
Yeah, go ahead.
waiting. Okay, so do you
We can see your screen now, David.
Okay, and you see presenter view or?
It's not in full screen mode, so
Okay,
Yeah, we see the thing on the side, you know, with the
yeah,
little
it would be
tiling.
good for you to
Yeah.
put it in full screen mode. But if you put it in presenter view, I think we'll see the presenter view, not the full screen. You can try it.
This will work too. I mean, if you have to use this is fine too.
Yeah, yeah. Under slideshow, David.
it's just because it's sharing on that screen. We need to get it sharing on this screen. And so,
Amber, are you there
Right.
by chance?
You,
you can also share different. I will tell you, if you do, yeah, let's do that.
Let's do that again. Okay.
Share screen. All right, so it looks like it's going to maybe if you, yeah, you can do screen 2, and then hit share. And then let's go ahead and have you hit that present.
There you go.
that's good.
That work?
You got your
Yes, you
screen
don't
up.
see
You
your
get
slides. You don't have
see-
the,
You see your sort of face page.
you don't have the PowerPoint up.
There you
There
go.
it is.
Now it's
Perfect.
good. You got it.
Good.
That-
Okay,
That's
great.
perfect.
I think, Richie, let's wait a while here because we got to get, people are going to just be keeping
Okay.
coming in.
Yep.
Yeah, the students ask tough questions too. Good ones.
Well, they're primed to do that. Richie has them in a course for a whole semester, doing nothing but reading your papers and trying to trick you a little bit. So. I think we'll just run over a little bit and, cut the lunch break down a little.
Okay.
Sounds good.
It would be nice if there was a way to get that beeping sound out when people join, because I know it's going to be like one of those attention tests, David, for you. A distractor,
Yeah. Well, I'm going to talk for a minute about distractors and working memory tasks. So.
Good. What do you think, Richie? You want to get started?
Yeah, I think we should get started.
Okay, let's do it.
Okay, so let me welcome everyone. And David, it is really an honor to have you with us today. For those who want to read about David's extraordinary accomplishments, please go to the Health Emotions website where each speaker's bios are presented. Very briefly, David is the chair of the Department of Psychiatry at the University of Pittsburgh. He holds the Thomas Detry Professor of Academic Psychiatry there, and he's been at Pittsburgh for many years. We won't hold it against David that he has received his degrees from the Ohio State University.
Are you sure?
David has published hundreds of papers, and he's made seminal contributions to our understanding, particularly of how prefrontal circuitry contributes to the cognitive dysfunctions in schizophrenia. This work is really groundbreaking, and he will share some of it with us today. So we're very fortunate to have him with us and welcome you to the symposium, David. Thank you.
Richie, thank you very much for that very generous introduction, and thanks to you and Ned for inviting me. It's been a great Symposium so far. Really enjoyed Walping and Tom's talks, and I hope to kind of complement the things that they mentioned by going through a strategy for understanding a neuropsychiatric illness, in this case, schizophrenia. And in reviewing this strategy, I hope that it will give you some ideas about ways that we can approach these complex psychiatric conditions that affect the most complex organ in the known universe. So schizophrenia is a common disorder, typically a chronic disorder associated with substantial comorbidities, and as a result, it's extremely costly in multiple dimensions. For the purposes of today in this session, I want to just point out that among the comorbidities of schizophrenia, depression is extremely common, such that individuals with schizophrenia have a risk of suicide, particularly early in the course of the illness, that rivals that of major depression. The context, though, of these mood disturbances in schizophrenia is always impairments in cognition. So, for example, cognitive impairments are present in individuals with schizophrenia years before the onset of mood symptoms or psychosis. As early as the first grade of school, people with schizophrenia underperform their peers and continue to have an increasing deficit in school performance well before they have any clinical symptoms. This cognitive impairment is highly prevalent in schizophrenia. Virtually everyone with the illness underperforms cognitively compared to what you would have predicted based upon their parental cognitive capacity. It's persistent across the course of the illness, and classic studies have shown that it is the best predictor of long-term functional outcome. How well someone with schizophrenia will manage their life is predicted not by how psychotic they are, but by their degree of cognitive impairment. And finally, keeping with this alliteration of P's, at least some of these cognitive impairments appear to be a product of impaired cortical network oscillations. So for example, here's a classic study from our study by Ray Cho, in which individuals were given a working memory task, a task that required them to remember a little bit of information for a short period of time in order to guide their response. During that task, they had an EEG recording, and during the delay period of the task, when they're maintaining the information that they need to guide their response, bilaterally over the frontal cortex, control subjects show an increase in oscillatory power of about 30 to 40 hertz, which is called the gamma band frequency. But you can see here, subjects with schizophrenia fail to show this increase in gamma band power. This observation has been replicated in antipsychotic naive subjects, and it may be a very informative biomarker because intracranial recordings in individuals doing the Sternberg task shows a strong relationship between working memory load and oscillation power. So in this task, over time, individuals are shown a set of 4 cards. The instructions are, remember what's on these cards, keep that information in mind while nothing is happening, and then when a probe card comes up, respond. Does this card match one of the 4 cards? And then forget all that information because the task is going to start over again. And what these intracranial recordings show is that gamma band power increases linearly with working memory load, stays high while the information is being retained, and then as soon as it is used and it's no longer necessary, oscillatory power drops. And in this same task, individuals with schizophrenia show a deficit in prefrontal gamma band power. So these findings suggest a possible way to begin to explore the disease process of schizophrenia. We can ask about how one aspect of the clinical syndrome, this impairment in working memory, and an apparent upstream pathophysiology of it, lower gamma band power, how might that arise from some disturbance in the prefrontal cortical circuit that is necessary for working memory and the oscillation at gamma frequency. So to address that question, though, in humans, as powerful as neuroimaging techniques are today, there is no way to get the cellular, synaptic, and molecular resolution required to understand a circuit without looking directly at the human brain. And of course, the only way we can do that is through postmortem human studies. But one of the challenges of those studies, as I would say, though, for any type of disease-related study, is that if we see something that differs in the brain of an individual with schizophrenia from an unaffected comparison subject, what does that actually represent? So we have the rule of the 5 critical C's in the lab. Does a given finding represent a potential upstream cause related to the etiology of the illness? Or might it be a downstream detrimental consequence of a cause? Or because the brain is highly plastic and driven to maintain homeostasis, could what looks abnormal actually be a compensatory response to a cause or consequence? Or, as I've already mentioned, there are lots of comorbid factors that accompany schizophrenia, so we have to make sure that what we see is actually part of the disease process. And finally, there's also the risk of confounds due to experimental limitations. So I'm going to focus on cause, consequence, and compensation for the rest of the talk, asking you to trust me that to the extent possible, we have excluded comorbidity and confounds as the basis for our observations. But just to illustrate 2 ways in which we've done that, we've used a model of antipsychotic administration to macaque monkeys to look at the effects of antipsychotics on prefrontal cortical circuitry. Here's some examples of studies in which we found positive findings, but the bottom line is that antipsychotic medications are associated, at least, with detectable effects on the brain in monkeys, but they don't appear to account for the data I'll present today. Likewise, we know that people with schizophrenia heavily use cannabis. So we developed a model for cannabis exposure in adolescent monkeys, did a number of studies looking at the effect of cannabis exposure on working memory. And what we observed is that repeated THC, the active ingredient of cannabis, does reversibly impair working memory function in monkeys. But again, in these monkeys, findings there don't seem to account for the data I'll present today. So back to how we're going to investigate this, we need to look into the neural circuitry of the human prefrontal cortex. So on the left, you have a lateral view of the human brain. The purple circle is the area of interest, the dorsolateral prefrontal cortex. If we were to make a coronal cut along the dotted line, you'd see the image shown on the right. Gray matter with underlying white matter. We're now going to look at that area in purple at higher magnification, looking at a Nissl-stained section on the left, where you can see from the top peel surface to the white matter, the 6 layers of the cortex defined by the relative size and packing density of their constituent neurons. And then if we look at layer 3 and turn up the magnification, you see the 2 main cell types of the cortex, excitatory pyramidal cells, which exist in about a 4-to-1 ratio with inhibitory GABA neurons. Now within layer 3 of the prefrontal cortex, a dominant circuit among these cells is formed between excitatory pyramidal cells and a particular type of inhibitory neuron called a parvalbumin-containing basket cell. The circuitry consists of recurrent axon collaterals from layer 3 pyramidal cells, 50% of which synapse on dendritic spines of other pyramidal cells, setting up a means for recurrent excitation. The other 50% go on to the dendrites of parvalbumin basket cells, which have wide arborizing axons, which innervate the cell body of pyramidal cells, providing feedback inhibition. This circuit appears to be critical for both working memory and gamma oscillations, as demonstrated in a series of studies in monkeys. So first, in classic studies from Patricia Goldman-Urquiche, neuronal firing during the delayed period of working memory tasks seems to arise from recurrent excitation among layer 3 pyramidal cells in the prefrontal cortex. Inhibition shapes this pyramidal cell activity during working memory tasks. As I've already mentioned, inhibitory parvalbumin basket cells are the predominant inhibitory cell class in that location. In that same location, studies from Earl Miller and others have shown that gamma oscillations, and especially gamma bursting during the delayed period of working memory tasks, predominates in layers 2 and 3. And then most recently in human prefrontal cortex, using an fMRI medium, layers 2 and 3 are preferentially active during the delayed period of working memory tasks. So this information all suggests that this circuit may be critically important for both generating gamma oscillations in order to perform working memory tasks, both of which are altered in schizophrenia, and hence perhaps this circuit is important in the illness. But how does the circuit generate gamma oscillations? Well, here's the same circuit in a different CAR2 fashion here. So 3 pyramidal cells, all of which innervate a PV basket neuron, which provides feedback inhibition to those cells. So you might imagine at some basal state, these 3 pyramidal cells are firing asynchronously. But when there's sufficient input to fire the PV basket cell, it spikes, and now it simultaneously inhibits all of the pyramidal cells. That inhibition then wears off together, allowing the pyramidal cells to spike synchronously, excite the PV basket cell, inhibit the pyramidal cells, and so on. So it's a beautiful mechanism for synchronizing the activity of pyramidal cells, doing so in a recurrent or oscillatory fashion, where the frequency of the oscillation is dependent upon the decay kinetics of the inhibition, which at this synapse tends to be about 25 milliseconds, providing a 40 hertz oscillation. So then when we think about schizophrenia, we can say, well, there's something wrong in the circuit. Where in the circuit is there a disturbance that leads to the impaired synchrony? Well, one possibility is it's in the excitation. So just using that as an open question, we can ask, is there a primary problem, primary in air quotes, in excitatory drive to layer 3 pyramidal cells? So to look at that, we need a mechanism to do it. And one of the ways to do that is to actually examine dendritic spines off of layer 3 pyramidal cells, because these spines are the principal site of excitatory inputs. So here's an electron micrograph for monkey prefrontal cortex, a parent dendrite. You can see the emergence of 2 dendritic spines. The spine heads are filled with actin filaments, which determine the morphology of the spine head. There's a postsynaptic density loaded with glutamatergic receptors, and then presynaptic to that is an axon terminal loaded with vesicles that contain glutamate. Spines serve as a proxy for a number of these excitatory synapses, and in a study that Lisa Glantz did a number of years ago, we used the classic Golgi technique to visualize dendritic spines in postmortem human cortex from comparison subjects and subjects with schizophrenia. And you can see qualitatively here in these extreme examples, there's a deficit in dendritic spine number in people with schizophrenia. And when Lisa and Nutan Fleury quantified this across cortical layers, they found that in deep layer 3, on average, there's about a 20% deficit in spine density in people with schizophrenia relative to both unaffected comparison and subjects with major depressive disorder. But this alteration is laminarly specific, not present on pyramidal neurons located in layers 5 or 6. So could this be evidence of deficient excitatory drive selectively on layer 3 pyramidal neurons in schizophrenia? Well, one reason to think that might be the case is that converging genetic evidence is consistent with a disturbance in dendritic spines in excitatory inputs in schizophrenia. So for example, common alleles associated with schizophrenia appear to be enriched for genes involved in glutamate neurotransmission. De novo mutations and rare variants are also enriched in loci that encode for postsynaptic proteins, and the strongest evidence is actually for proteins that regulate the actin filament dynamics that I've already illustrated for you are essential for dendritic spines. A study from the Broad Institute that got a lot of attention showed that a specific locus, complement component 4, that's strongly associated with schizophrenia, regulates developmental pruning of dendritic spines. So all of this evidence fits together with the idea that there may be a genetic basis for a primary disturbance in dendritic spines in schizophrenia. But if that's the case, why would the spine deficit be most prominent and potentially even selective for layer 3 pyramidal cells and not affect pyramidal cells in layers 5 and 6? Well, one possibility is that genetic liability could be moderated by cell type specific patterns of gene expression. Just to give you one example of a system that could account for that. So we found that expression levels of the Rho GTPase CDC42, which is a positive regulator of actin dynamics and spine number. So it increases spine number, and it is also involved in increasing spine size during plasticity. This particular transcript is lower in layer 3 pyramidal neurons in schizophrenia, and it's strongly correlated with spine deficits only in layer 3 pyramidal neurons. On the other hand, negative regulators of actin, such as CDC42 effector proteins, are actually upregulated in layer 3 pyramidal neurons in schizophrenia, and these gene products are preferentially expressed in layer 3. So together, these ideas of reciprocal alterations in positive and negative regulators of actin selectively in layer 3 pyramidal neurons could potentially account for a cell type specific dendritic spine deficit in these cells. So just to go back to this model of the disease, I've suggested to you that there is a component of the clinical syndrome, impairment in working memory, that's associated with a potential biomarker, lower gamma oscillation power. And on the other side of the disease process, we have genetic liabilities in actin regulation and cell type specific disturbances in a particular signaling pathway that could converge on producing fewer dendritic spines on layer 3 pyramidal cells in the circuit that is important for generating gamma oscillation power. So this gets us to the point where we can make some postulates and then test those through predictions. So the postulates, just to repeat what I've said, is that schizophrenia is associated with genetic and transcriptome alterations in the regulation of actin that impairs the formation and maintenance of dendritic spines. These are most prominent in layer 3 pyramidal cells due to cell type specific alterations in gene products, and that fewer spines and glutamatergic synapses would be expected to reduce excitatory input to layer 3 pyramidal cells because that's where all the excitatory input comes from. Now, if those postulates are correct, then the prediction would be that layer 3 pyramidal cells are hypoactive in schizophrenia because they have less excitatory input, and because excitation or activity of a neuron is the principal driver for mitochondrial energy production, they would have less drive to make energy. So can we test these ideas by asking, are markers of neuronal activity and energy production lower potentially as a consequence of fewer dendritic spines on layer 3 pyramidal neurons? So one way to look at that is to look at genes that are expressed in an activity-dependent fashion. A classic example of one of those is neuronal activity-regulated contraxin 2, or NPTX2. This gene is expressed by pyramidal cells in response to neuronal activity, and it's particularly interesting because it's then secreted from presynaptic axon terminals onto glutamatergic synapses at PV neurons. So it's right active in the circuit we're interested in. In this particular study, Sohei Kimoto looked at the levels of NPTX2 in 62 pairs of schizophrenia and comparison subjects. Let me just explain how the data are presented here because I'll show you other plots like this. So the x-axis is the NPTX2 level in every comparison subject, the y-axis in every schizophrenia subject. Each dot represents a matched pair of subjects. This line is not a correlation line, it's the unity line. So any value below that line means that that schizophrenia subject has a lower level of NPTX2 than they should relative to their age, sex, etc. matched comparison subject. So about 85% of schizophrenia subjects have lower NPTX2. Similar things are true for other activity-dependent transcripts. So the first test of the hypothesis is yes, there does seem to be evidence of lower activity in layer 3 pyramidal neurons. Well, what about energy production? Well, to test that idea, we used what was then a cutting-edge technique, laser microdissection, of individual layer 3 pyramidal cells from postmortem human cortex. So here's a Nissl stained pyramidal cell, here's the laser cutting it out. We collect these, we pull them, we subject them to RNA profiling. And what popped out of this initial study that Dominique Ariane conducted was that the gene pathways that were most significantly and profoundly altered in layer 3 pyramidal neurons in people with schizophrenia were all focused on genes that are involved in mitochondrial energy production, all markedly underexpressed. And this seemed to show when we looked at individual genes with PCR in cell populations or in total gray matter, preferentially expressed in pyramidal cells. So these same gene products much greater deficit in schizophrenia selectively in layer 3 pyramidal cells than in total gray matter. So we can go back to our postulates and predictions and suggest now in layer 3 of the prefrontal cortex, the quote cause is a lower number of pyramidal neuron dendritic spines predicted to result in lower excitatory drive to layer 3 pyramidal neurons. As a consequence, we have indices of net neural activity that are lower in this circuit. And so we can make a prediction that due to homeostatic synaptic plasticity mechanisms, all of which are designed to keep the brain at an appropriate level of activity, that in response to this deficit in excitation, there would be multiple pre- and postsynaptic presumptive compensations in which PV basket cell inhibition of layer 3 pyramidal neurons would be downregulated. And this nicely follows from experimental evidence that if you overexpress a potassium channel that inactivates pyramidal cells in mouse visual cortex, you see a homeostatic downregulation of PV neuron-mediated inhibition. So the question is then, is at this synapse, the PV cell input to pyramidal cell bodies, is there evidence that inhibition is downregulated both pre- and postsynaptically to compensate for lower levels of activity? So I'm going to briefly show you 4 studies in this cartoon. So here's a PV basket cell terminal, pyramidal cell body. These are GABA A receptors. In this terminal, the synthesizing enzyme for GABA, GAD67, responsible for most GABA synthesis. We can measure the protein levels of the terminal. And what we observed here was lower levels of this protein in people with schizophrenia, consistent with lower GABA synthesis and potentially less synaptic strength in work that Allison Curley did. Postsynaptically, these GABA A receptors contain an alpha-1 subunit. Joe Glausier quantified the level of GABA A alpha-1 mRNA. It's lower in people with schizophrenia. So a reduction in the number of receptors and lower synaptic strength. When GABA binds to these GABA A receptors, fluoride flows in and hyperpolarizes the cell. What Dominique Ariane observed was that intracellular levels of fluoride were basally increased due to a shift in chloride transporters, suggesting that there will be less chloride influx and less hyperpolarization when synaptic GABA neurotransmission occurs. And then finally, the mu-opioid receptor has its one side of localization on the axon terminals of PV basket cells, where it suppresses GABA release. And David Volk demonstrated that the levels of the mu-opioid receptor are actually increased on these terminals in schizophrenia, suggesting greater suppression of GABA release. So 4 separate changes, 2 presynaptic, 2 postsynaptic, but all converge on less GABA neurotransmission at this synapse. So our provisional interpretation is that the cause of the working memory relevant circuit dysfunction in the prefrontal cortex in schizophrenia is a cell type specific deficit in dendritic spine number, resulting in lower excitatory drive to layer 3 pyramidal neurons. As a consequence, net neural activity is reduced in layer 3 circuitry, and then homeostatic processes induce molecular mechanisms producing multiple compensations that lower PV cell basket inhibition of layer 3 pyramidal neurons. But there's a caveat. I've been telling you a story. And one of the things that we know about stories, according to Daniel Kahneman, is that our confidence in an explanatory story rests not in an evaluation of the evidence and its quality, particularly when it's given in a talk like this, but rather in the coherence of the story. You know, does it hang together? And it's much easier to construct a coherent story when there are fewer pieces to the puzzle. So what happens to the story I've told you if we add more pieces to the puzzle? Well, one piece that we'd like to add is, well, is there any evidence that these alterations in layer 3 pyramidal cells, are they actually associated with the cognitive deficits that we've been trying to understand? Well, of course, very difficult to do cognitive testing in postmortem human brain, but we can do some comparative studies to try to assess that. One comparison that we can make is to people with bipolar disorder who have many of the features of the people with schizophrenia have. They certainly have a chronic, severe psychiatric illness. But as shown in this meta-analysis, they don't perform as poorly on working memory tasks as do people with schizophrenia. In fact, people with schizophrenia have on average about a 0.5 greater deficit effect size in working memory function. So we thought, well, let's look at layer 3 pyramidal neurons, mitochondrial gene expression, head-to-head in people with schizophrenia and bipolar disorder. So in this study that Joe Glaser and John Enright conducted, we looked at about 1,000 genes that have been attributed or associated with mitochondrial function, and found, as you can see in these volcano plots, that there are a number of genes that are significantly either under or overexpressed in schizophrenia, many fewer in people with bipolar disorder. Here's the aggregate number. So indicates that there is a major difference in people with schizophrenia in this mitochondrial deficit in layer 3 pyramidal neurons, which would be consistent with, certainly not proof of, the basis for the greater cognitive impairment in schizophrenia than in bipolar disorder. But I would note, when we look at the test statistic for all of these genes between people with schizophrenia and bipolar disorder, there is a much greater spread of the test statistic in schizophrenia than in bipolar disorder, but they're strongly correlated, suggesting that people with bipolar disorder do have somewhat of a signature similar to people with schizophrenia. It's just much less severe. And potentially that could serve as both a substrate for the working memory impairments in people with bipolar disorder, but also explain why they're not nearly as bad as they are in people with schizophrenia. Okay, so back to this caveat. Can we add more pieces to the puzzle in terms of cortical circuitry? Well, I'm going to just focus on two other pieces that I want to review to kind of further test the idea. And that is that we know that there are other sources of inhibitory regulation of layer 3 pyramidal cells. So here's the parvalbumin basket cell that I was talking to you about, its input onto the cell body of pyramidal neurons. There's another important cell class that contains the neuropeptide somatostatin that innervates the distal dendrites of pyramidal neurons. Somatostatin cells may be particularly interesting to look at because Josh Gordon's work in this paper by Abbas et al. Showed that silencing of somatostatin neurons in rodents impairs working memory function. And we had previously shown that there are marked deficits in the levels of somatostatin mRNA in people with schizophrenia, on average about a 40% deficit. And here in this paired subject study, you can see that most of the subjects with schizophrenia have a lower level of somatostatin message in the prefrontal cortex than their matched comparison subject. Why this is important is that current ideas suggest that while the parvalbumin cells are critical for feedback inhibition of the circuit in layer 3, somatostatin cells play a critical role in laterally inhibiting other pyramidal cells that should not be activated during the working memory task. And where that may fit into the nature of working memory tasks are, here's a classic visual spatial working memory task where a human or a monkey is fixates on a central cue. Then when that disappears, a cue signal comes up. This is the memoranda. Remember this spatial location. Then there's a delay period when there's nothing to look at. Then a distractor pops up. Similar cue, but in a different location. And then the individual needs to respond, and a correct response is, of course, to look in the location where the cue was. Well, what we think somatostatin cells are doing is suppressing the activity of pyramidal cells that would be responding to the distractor. And people with schizophrenia, one of their impairments on working memory tasks is they're more liable to show impaired performance when distractors are introduced. So that's one piece that we can add to the puzzle. What's going on in somatostatin cells in schizophrenia? But another piece of the puzzle is to recognize that working memory is not solely mediated by the prefrontal cortex, but it's dependent upon a distributed cortical network. So for visual spatial working memory, information comes into primary visual cortex, goes to visual association cortex, is ultimately relayed to the posterior parietal cortex, and then from there to the dorsolateral prefrontal cortex. So we wanted to look at these four regions of this distributed network in the same subject with schizophrenia and ask, what's the nature of the GABAergic parvalbumin and somatocell alterations in these 4 regions. So we're going to look at what happens to the story if we add somatostatin neurons and other cortical regions to the puzzle. So the first study was just to look at markers of GABA cell types in these 4 regions of the human visual spatial working memory network. We looked at 8 GABA neuron transcripts that are specific to certain GABA neuron subtypes, shared by a subset of subtypes, or are common to all GABA neurons. What we observed in unaffected comparison subjects is that there are marked regional differences in the nature of this circuit. So for GAD67, which is present in virtually all GABA neurons, very little differences in the level of GAD67 across regions from the back of the brain to the front of the brain. Likewise, for a third population of GABA neurons, VIP. But notice there are marked regional differences in the presence of parvalbumin, major decline from the back of the brain to the front. Oops. And for, back a second here. Sorry. And for somatostatin, exactly the opposite pattern. So very different. But when you look at what happens in schizophrenia, there's a highly conserved effect across regions. So what's plotted here for each of these 4 regions is the ratio of schizophrenia to comparison subject for each transcript. So a value below 1 means less than schizophrenia, a value above 1 means higher levels of schizophrenia. Several points to point out. First, this finding replicates what I've already shown you in the prefrontal cortex. Low levels of somatostatin, of parvalbumin, and an upregulation of the mu-opioid receptor. But when you look across these regions, what you can see is that the rank order of the disease effect is preserved. The greatest deficit is somatostatin, next in parvalbumin, the greatest upregulation, mu-opioid receptor. But when you look across regions, you'll also notice that the magnitude of the effect seems quite similar for the 3 posterior regions, but it's much smaller in the prefrontal cortex, exactly the opposite of what we initially expected. So does this reflect differences in the vulnerability or potentially in the compensatory capacity of the local working memory circuit across regions? Well, to explore that further, we wanted to look at things in a different way, and now we wanted to look specifically not just at the cell bodies and at transcripts, but at proteins in axon terminals. So we conducted a study led by Ken Fish, in which we looked at layer 3 parvalbumin basket cell axon terminals and measured proteins in those terminals, including GAD67 and GAD65, the 2 enzymes that are responsible for GABA synthesis, and together they provide an index of the strength of GABA neurotransmission. Did this in the same regions, the same studies of the subject same subjects of the study I just showed you. The technique that's used here enabled us to visualize selectively PV basket cell terminals in postmortem human brain. So in this image, it's labeled for VGAP, which is a vesicular GABA protein present, GABA transporter protein present in all GABA terminals. So here you can see with this arrow, a PV or a VGAP positive terminal on an unlabeled pyramidal cell body. This same terminal contains PV, it contains GAD67, it contains GAD65. So it is the classic definition of a parvalbumin basket cell terminal. It contains all the right proteins, and it is immediately opposed to the cell body of a pyramidal neuron. So using that technique, we again look first just at the comparison subjects, and very similar to what we observed for parvalbumin message, indices of parvalbumin axon terminals, whether the terminal number or PV protein levels, decline from the back of the brain to the front. And in schizophrenia, we looked at an aggregate measure of synaptic strength in these terminals, which was the sum of the number of terminals and of GAD proteins in these. and found, similar to the mRNA study, that there is a deficit in PV basket cell terminal GAD levels across all regions. But just as in the message study, the deficit, the effect size of the illness, is only about a third of that in the prefrontal cortex as it was in the 3 caudal region. So this leads us to have to reconsider the story, and now leads us to provisional interpretation. So first, the observation is that similar to somatostatin, parvalbumin, and GAD67 mRNA levels, the magnitude of the disease effect on this composite GABA transmission measure in the prefrontal cortex was only about 40% of that in the other regions of the working memory network. But in functional neuroimaging studies of people with schizophrenia doing a working memory task, the disease effect is always much larger in the prefrontal cortex. They fail to activate the prefrontal cortex to a greater degree than they fail to activate more caudal regions. So what these findings could suggest is that there is in actuality a lower compensatory capacity of the GABAergic components of the local working memory circuit in the prefrontal cortex. That is, that the reason that the prefrontal cortex appears so preferentially impaired in schizophrenia is both because it is a home to the disease pathology, but also because there's something about the circuitry there that doesn't allow it to compensate to the same degree that the circuit in more posterior regions compensates. So if inhibition downregulates to compensate for deficient capacity of layer 3 pyramidal neurons to be activated, so the premise I've been showing you earlier in the talk, then insufficient compensation in the prefrontal cortex would contribute to its more prominent altered activation during working memory tasks. So from our view, this is kind of a disease process of schizophrenia. It's a testable model in progress. So the components that I showed you earlier are illustrated here, but now what we think the pathological entity involves is this combination of a genetic cell type driven deficit in dendritic spines that results in lower activity, but also is associated with a deficient compensation in the layer 3 circuit. So that explains why one of the challenges that we had earlier on, well, if the circuit is compensating by downregulating inhibition, why isn't it normalized? And it appears to be because the compensation, for reasons we have yet to discover, in the prefrontal cortex is less adequate than it is in more posterior regions. So why is this kind of model important? Well, I think it's helpful from the perspective of it tells us something about where to target prevention and treatment. So I don't think, given the complex genetic and environmental risk for schizophrenia, that we'll ever have a primary prevention approach. But knowing something about the pathogenesis may give us ideas in individuals who are at risk about how to preemptively intervene and normalize the capacity of these layer 3 pyramidal cells to form and maintain dendritic spines, perhaps using the types of strategies that Guoping illustrated in his talk at the beginning of the day. But knowing about the nature of this compensation also suggests that in terms of treatment, rather than just trying to attack the pathology, we may be able to design therapies that would preferentially augment the compensation selectively in the prefrontal cortex. And so what I showed you earlier about marked regional differences in what the canonical cortical circuit is, that the prefrontal cortex is very different from the visual cortex, provides some hope that we may in fact actually be able to develop circuit-specific means of augmenting compensatory capacity and enable the brain to accomplish what it was actually trying to do in the face of the pathology. And then, of course, knowing the association of those with this pathophysiology gives us a potential biomarker to use in early stage trials to ask whether our intervention to enhance compensation does in fact increase gamma band power well before we could actually measure whether it has an impact on the clinical syndrome. So let me stop at that point in acknowledging the many individuals who were key to this work. I think I mentioned most of the key collaborators listed here during the course of the talk. Also want to acknowledge and thank our key brain tissue donation team currently supported by an NIH contract to acquire brain tissue specimens and all the clinical information needed to be able to conduct these studies. And especially want to acknowledge and thank the many family members who at a very difficult time in their lives generously gave consent for brain tissue donation and then continued to work with us through exhaustive diagnostic interviews, and then of course support from the NIH, multiple foundations. So thank you very much for the opportunity to present today, and I look forward to a set of challenging questions from the students.
Great talk, David. Thank you very much. Really, really quite interesting.
Yeah, thank you so much. And it's just so inspiring to see this painstaking work. And I just really appreciate your acknowledgement of the sensitive and complex nature of how you acquire these samples that you discussed at the end. So thank you for sharing that. Our questions will be led by Brooke, who is one of our trainees in our T32. And again, she will be presenting questions aggregated from a group of trainees. So Brooke.
Hi, thank you so much, Dr. Lewis, for your presentation. We really appreciate it. And yeah, we've had a lot of reflection this semester on your work, so it's really exciting to get to ask you some of these questions. So we have 4 questions for you. I don't know how many we could get through. The first one is a little more specific, and the other 3 are a little bit more general. So the first is from the Glaucier et al. 2020 paper, which you discussed a little bit in your talk. Based on your findings with the alterations in the mitochondrial pathways associated with schizophrenia, but not as much with bipolar disorder, what might you say are the implications of this study in better understanding the role of the mitochondria in schizophrenia and its symptoms? In other words, how do these dysfunctions in the mitochondria affect the overall symptomatology found in schizophrenia patients?
Yeah, thanks, Brooke. That's a great question. And I think it's really important to clarify that what we think our findings show is not that there is a primary mitochondrial disturbance, but that the mitochondria are responding appropriately in downregulating energy production in cells that are not receiving a lot of synaptic input and hence are firing less. And so we don't see evidence, so one of the tests of that is in primary mitochondrial disorders. If you look at mitochondrial related transcripts, they tend to no longer have coordinated expression. What we saw in the Glaucier study was that the expression of all these transcripts was as strongly correlated in people with schizophrenia as it was in comparison subjects. Second reason for thinking this is not a mitochondrial abnormality is that it's indirect, but in the genetic studies, there really haven't been any hits based on de novo mutations or common variants that are at least in the loci that contain key mitochondrial genes. The third reason, I think this was just more preliminary, but it's work that Jill is doing now, is actually looking at mitochondrial structure by EM. And that structure is consistent with changes that mitochondria normally undergo when they're dysquiescent, not when they are pathological. So we think this is an important signal, but it's a signal that's reflective of what the brain is doing in response to an upstream effect, not because there is a primary abnormality in mitochondria. So that's good from the perspective of thinking therapeutically, because what it may mean is that, well, if we could enhance the compensation so there's less inhibition, or if we could enhance the excitation, that the mitochondria would be well equipped to produce the energy needed for the cells to engage in normal levels of synaptic activity.
That was really helpful clarification, and thank you for expanding on that. So the second question we had for you is, in what way could your work be used to better diagnose or treat specific disorders within the schizophrenia spectrum or clusters of symptoms? Can any of the work you've been doing be used to subtype schizophrenia into more biologically meaningful categories that have implications for differential intervention?
Yeah, it's both great questions. So as I kind of alluded to at the end, what we hope is that that we can develop a robust correlation between an objective, quantifiable index of brain dysfunction and schizophrenia, in this case, working memory. So that gets us, what we struggle with clinically, of course, is that we're dependent upon what people tell us about how they're feeling or what we can observe. And those are at best qualitative and frequently surely subjective. So working memory is a type of task that's objective and quantifiable. I also think it's an important task because I think it really indexes one of the key clinical features of schizophrenia, which is loose associations. So you can imagine, and if we have time, I'll diverge into what was a very influential experience in my life. When I was a third-year medical student, my first rotation, my first patient when I did my psychiatry rotation was an 18-year-old high school student who'd been admitted the night before for a new onset of psychosis. And when I went in to interview him the next day, by myself as a third-year medical student, not knowing much, I said to him, "What brought you to the hospital?" Which was the same question that I'd asked on general surgery. I'd learned not to ask it on OBGYN because women thought, if you can't tell why I'm here in the delivery room. But when I asked Mark this question, he said, "A police car." It was a Buick. Buicks are made by General Motors. General Motors is the largest car manufacturer in the world. The world's going to end in 2010 according to the Mayan calendar. The Mayans lived in Mexico. When I went to Mexico, I got really sick. So this is a classic, and I wrote that down because it was so distinctive of a representation of loose association. He could not keep the context at all in mind of the question that I asked. And in fact, if you looked at those sentences, the stimulus for one sentence was the last thing that he said in the previous sentence. So he had no internal representation that was guiding it. Well, it took me a long time and a number of years later before I reflected back on that and realized, that's actually an extreme example of a working memory impairment. The inability to keep in mind context in order to guide thought or behavior. So that's why I think working memory is a really useful thing for us to be studying. Not only is it objective and quantifiable, but it also may be an index of lucid associations. And in fact, Cam Carter has shown that it nicely correlates with general disorganization. Okay, so, but to be able to attack that, it's really useful to know if we have a biomarker index of it. And that's why I think our gamma oscillations are potentially important. Now, there's a lot of variability in gamma, so it may not turn out ultimately to be useful. But if we imagine that it was, then that tells us, well, this is possible. If we make an intervention, we have perhaps a quick way to determine whether it's going to be useful in people by seeing whether it changes the physiology or it budges the physiology, even if we can't yet see it impact the clinical syndrome. So it's a long-winded segue to the question, but I hope I got at least part of the question. I think I may, due to limited capacity in working memory, I may have forgotten the other part of the question. Or I served as a distractor for myself, I guess.
So the other part of the question was, can any of the work you've been doing be used to subtype schizophrenia into more biological meaningful? Great.
So one of the things that has emerged from our studies is that there do seem to be, and we've reported this, David Volk has reported it in two different papers, a subtype of people with schizophrenia who don't seem to have any of this compensatory GABAergic alterations. So when you do a cluster analysis, they cluster with unaffected comparison subjects. We were initially confused by that because we thought, oh, well, guys, but they're sick. How can, if this is important, how can that be? But now when we think of this as an inadequate compensation, it is those people may be the sickest because they've shown the least degree of compensation. So very early, it doesn't correlate with any, classic clinical subtypes, paranoid, undifferentiated, et cetera. But it may at least be the kind of a substrate for some biological basis for subclassing.
That's really interesting. Yeah, thank you for addressing all of that and giving a lot of background for your answer. So the third question we had was, one of the major limitations in studying schizophrenia in humans is the inability to manipulate medication intake other than clinical trials specifically to test certain medications. And there's a wide variety of possible medications these individuals can be on. How do you see the future of neuroscience work in schizophrenia combating this limitation?
Yeah, so I think that this gets to a very important point that one of the, I think, a potential reason why a number of promising compounds failed over the last decade is that they were all tried in people who had chronic schizophrenia, who were still on antipsychotic medications, who'd had 20, 30 years of impaired cognitive function. These are drugs to try to improve cognition, and now I think that the recognition of that is such that there's at least an attempt to try to conduct studies of novel cognitive enhancing agents in people who are in their first episode or early episode, or ultimately, if we get better at predicting who's really at clinical high risk, at individuals who have some cognitive impairment who haven't yet become psychotic. So I think changing the whole paradigm about how we assess drugs that have strong scientific rationale and that have evidence that they move the relevant biomarker in the desired direction, how we actually assess them for clinical efficacy may really require this paradigm shift in the subjects that we select for study.
Okay, thank you. That's also really helpful. The final question that we had kind of has a few parts to it, but a lot of the work you're doing is dependent on access to the brain bank, which you mentioned during your presentation. To what extent is there range of patient severity represented in the brain bank? And to what extent is the schizophrenia spectrum represented? And then could you also tell us a little bit more about the establishment of the brain bank itself, as well as what information is available that might help phenotype patients in a more granular way?
Sure, all great questions. So in regard to the first question, the way that our brain bank operates is it's through a medical examiner office, which has several strengths, one of which is that patients that we recover brain tissue specimen from have all died suddenly, unexpectedly, out of the hospital. And the recovery time between when the event occurred and we're able to receive and preserve the brain is less than 24 hours. So that means we don't have to worry about agonal state events, somebody who had pneumonia and progressively became hypoxic before they died. We don't have to be concerned about the effects of medical interventions that occur that can, when someone's trying to be saved. And it means that the effects that occur post-mortem at least are relatively constrained. What it does mean is that our sample is somewhat biased towards people who die under those conditions. So about 2/3 of our sample are males of the whole sample. That's part because males are more prone to do things that are going to get them to die suddenly, unexpectedly out of hospital than are women. It also has some effect on the age range. So, but for the people with schizophrenia, it pretty much means that we capture the spectrum. So we have people who, in the data I showed you today, had only been ill for a year. They were 18, 19, 20 years of age, and people who'd been ill for 30 or 40 years who were 60, 65 years of age. That's pretty much our cutoff. One of the advantages of that is, of course, it enables us to look both at the impact of age, but also, to co-factor out age and look at duration of illness, and to see to what extent that affects these measures. And so one of the things I didn't mention is that everything that I've shown you today that we've looked at is there in people. For example, the somatostatin finding, the difference between an unaffected comparison subject is the same in somebody who's 20 years old as it is in somebody who's 60 years old, even though normally there's a marked decline in somatostatin levels with age. Okay. So we're able to kind of control for things like that. To your second question, we are now part of what's called the NIH Neurobiobank. So we're supported by a contract from the NIH, where there are 5 other sites. Anyone in the country, anyone in the world can go to the Neurobiobank portal and look at the available specimens and develop a cohort for study. In fact, anybody who's listening who's interested in that, I would encourage you to do that because it's a great resource. For our samples, the way we clinically characterize them, as I alluded to at the end, is by developing a relationship with the next of kin and other family members. So it begins when one of our clinical psychologists calls for consent for brain tissue donation. We then have a structured interview, which at a time that the family is ready for, we conduct with them. It's a skid interview. There's all sorts of other assessments that we would do in living subjects. We get all the medical records. We get school and police records. We look on social media. We put all that information into a life history summary and then present that to an independent panel of research clinicians who render consensus DSM diagnoses and their level of comfort of those diagnoses. Long-winded answer, but it was a 3-part question. So.
It was. Thank you. It seems like so much has to go in to be able to get that going. And then just a follow-up question to that, do you have any recommendations for establishment of a brain bank for other areas of psychological research? For example, a well-being brain bank?
That, that is a great, yeah, and we actually, so for our unaffected comparison subjects, we do the same interview with the family that we do with people with suspected psychiatric illness in order to confirm the absence of it. But we have not looked at it from the standpoint of to what extent are we assessing well-being or resilience or factors like that. And we may not, we probably have some data that would be relevant to that, but there's probably a lot of things that we could be asking that we're not. So I'd love to have a follow-up conversation about how that might be done, and we could potentially influence the entire NIH Neurobiobank.
I'll follow up with you on that, David.
Okay.
I've got some ideas.
Great.
Yeah, that's great. Well, thank you, Brooke, for the great questions. And we are running a little past our time. And David, I hope you can join us for our general discussion at the end of the day.
Yeah,
so,
definitely be there. Yeah.
Great. We'll have more of a chance to ask additional questions then. So we're scheduled to take a break now for lunch for those of us in the lunch-related time zones now. And we will resume at 1 o'clock Central Time with talk by Tracy Bale. And Nick, do you have any final words you'd like to
No, just want to thank David again. Just a really exciting talk, David. And I think parsing the compensatory from the sort of more primary pathophysiological processes and guiding us to think about treatments that are related to those different elements is sort of a good generic framework to be thinking about in relation to a lot of different things that we think about in psychiatry and new treatments. So thanks again. Really cool.
Thank you very much.
Really appreciate it. Thank you, and see you all back at 1.
Okay, take care.
Bye-bye.
Bye-bye.
We're going to, before we go, we're going to go to the other platform at 1. I think let's just remind everybody about that. So, yeah.